is having some issues. It's said, I think. And is Kieran in the lesson? Is he in the lesson? It's not there.
So I am in the lesson. Sorry, my mic wasn't working.
Okay, so what did I say? The passing on of genetic information, genes, genetic information from one generation to another. Passing on of genetic information from one generation to another. To another. That's what we're talking about here. The passing on of information from one generation to another. But my question now is, how does that happen? How does that happen? So what happens is that inside the cells, your human cells, you have your nucleus, which you know very well. Inside every human cell has got a nucleus. And inside or animal cell, inside the nucleus, we have what are called chromosomes. Chromosomes, they are the ones that are responsible for what we are talking about here. Clear? Inside the nucleus, every human cell or animal cell, we've got what are called chromosomes. They are the ones that are responsible for the passing on of genetic information from one generation to another. Clear? And what you need to know that a normal human cell has got how many chromosomes? In fact, we can actually say all human cells.
Sir.
Except,
You said,
yes.
So you said chromosomes are what?
We remember the definition. We said inheritance is the passing on of genetic information from one generation to another. And I said for that to happen, it happens through the cells. When the cell division, it happens through the cells. I'm saying inside every human cell, we've got a nucleus. And inside the nucleus, we have got what are called chromosomes. We haven't talked about the definition here. We have what are called chromosomes. That's what I said. Then I said every human cell, except the sex cells, except your male gametes, every human cell except the sex cells has 46 chromosomes. Clear? 46 chromosomes. So every human cell, any other cell that is not a sex cell has got 46 chromosomes. Are we together on that?
Yes, sir.
Yes, question? Oh, yeah, agreeing. Is that clear on that? So in terms of your chromosomes, okay, let me just try to draw a diagram here. Your chromosomes, I think you've seen many textbooks where you've seen something that is coiled, can be in the very different, maybe different shapes. Okay. So your chromosome is just a long thread of DNA, yeah? Clear? Are we together? It's a long thread-like structure of DNA. The chromosome is a long thread-like structure of DNA. Clear? Are we together? Is that making sense?
Sir, I couldn't hear you.
Okay, I said a chromosome is a thread-like structure, a long thread-like structure of DNA. A long thread-like structure of DNA is what we call a chromosome. Are we together? Which carries genetic information? Which carries genetic information? Which carries genetic information in form of genes, which carries genetic information in form of genes. In form of genes. So you find that your chromosome is divided into sections like this. I'm talking of sections here. So these sections are actually what we call genes. So a gene is a gene as a definition is a section of DNA. A gene is a section of DNA. A gene is a section of DNA that codes for a specific protein. That codes, you need to know this definition, for a specific protein. Is a thread-like structure. Sorry, it's a section of DNA that codes for a specific protein. Are we together? A gene. Clear? Then we've got an allele, but this will make sense when you look at the diagrams just now. It's just a different version of the same gene. A different version of the same gene. Can be gene for toe or what? A different version of the same gene. Are we together? So we've just started with the basic definitions that you need to know, that are important for your syllabus. Now let's look at quickly the formation, protein formation or protein synthesis. How proteins are made. Protein synthesis. Protein synthesis. Protein synthesis. So remember we talked about in the nucleus, we said there are chromosomes. Okay, if I draw a human cell here, I said in the nucleus, we've got chromosomes. So the first thing that happens, was it's talking of protein synthesis here, is that the DNA inside the chromosomes, the DNA, you don't have to write it down, I simply need to explain. What happens is that a copy of the DNA is made first. A copy of the same DNA is made inside the nucleus. Then we have a message carrier, clear, called the mRNA. We have got a message carrier molecule. So this message carrier is the one that attaches itself to this DNA there, and then transfers this copy to the cytoplasm there via the ribosome. Are we together? And now the ribosome is the one that now assembles the amino acids into different protein structures. Is that clear? So number one is Sarah Beck in the lesson. Now you can write now. So number one, now I'm summarizing. So you can write one. A copy of the DNA. A copy of the DNA is made in the nucleus. A copy of the DNA is made in the nucleus. The mRNA, small letter M, then the rest, capital letters, the mRNA attaches itself to the copy. Attaches itself to the copy of the DNA and carries it to the cytoplasm. and carries it to the cytoplasm. Clear? And carries it to the cytoplasm. The next point is that the mRNA passes through the ribosomes, ribosomes.
Wait, and carries?
Yes, slow. And carries the DNA to the
Right.
cytoplasm. Carries the DNA to the cytoplasm. That's what I said.
I didn't hear you, Mr.
Okay, carries the DNA to the cytoplasm. Then I said
You're
after,
breaking up.
maybe it's your energy, carries the DNA to the cytoplasm.
Yeah.
Okay. Then I said the next point is that the mRNA passes through the ribosomes. That's the next point. After that, the mRNA passes through the ribosomes. Clear? Or not clear?
Yeah, it's clear.
Through the ribosomes. Okay, hold on. Then the next point is that the ribosomes assembles the amino acids into different protein molecules. The DNA, sorry, the ribosome assembles amino acids into different different protein molecules, depending on their bases. Depending on their bases. Are we together? Yes. Question?
Into different protein molecules.
Yes, into different protein structures, molecules.
Just for protein molecules.
So protein molecules, you have so many different protein molecules, depending on their basis. Do you agree?
Yeah.
Okay. There are 4 different bases that exist in a DNA. There are 4 different bases that exist in a DNA. You are not required to know the names. You're simply required to know the bases, but I'll give you the names. 4 different bases. There is A, then there's base T, there is base C, there is base G. So I said for the O level, the names are not important. It's what is important is just to know the initials. I mean, are we together? What's important to know is just to know that there's base A, base T, base C, and base G. Second point, A is complementary to T. That's what is very important now. A is complementary, which means A always A always forms a link with T. Yeah? So A is complementary to T, and C is complementary to the G base pairing. Clear? I said A is complementary to T, and C is complementary to G base pairing. A is always complementary to T, and C is complementary to G. This is known as base pairing rule. This is known as base pairing rule. This is known as base pairing rule. I hope it's making sense. This is known as base pairing rule.
So A is complementary to T and C is complementary to G, isn't
I
it?
said A is complementary to T and G, series C, yes, is complementary to G.
Okay.
Can I show you something?
Are we together? Or
Yeah.
it's not clear? Clear?
Yes, sir.
Yeah.
Okay. I said that is known as the best pairing rule. But now we need to be careful now when it comes to the RMNA. Yeah? This is true for every DNA that you can think of, except the mRNA. Yeah? This is the one that has got an exception. Huh? Clear? Are we together? What's the difference between, who knows what's the difference with an mRNA? We talked about it before. What's the difference? What's the difference? What is not existing? So you can write that in mRNA, there is no base here. No base. No base T. Clear. The T is replaced by what? T is replaced by? What replaces T? U. T is replaced by U. Are we together? T is replaced by U. Making sense?
Yeah.
So now I want to write something. Let's say we've got a protein here, A. Then we are told that this is an mRNA. We are told that this is CGT. A. Okay, hold on. Let's say this is molecule 1, or this is a, then this is B. Then after B, we want to look at if it was a mRNA, yeah, like this. So I have this base sequence. I've got A, C, G, T. Then I've got G, G, T, A, something like this. Now, who can tell me the base pairing here? This is just another normal protein. What would be the base pairing here? What should I write there?
C.
C, that's what you said. Because we said G is complementary to C. So now this will pair to a what? To a G. But this is not an mRNA. Then T will pair to a what?
A
This will pair to what? To T.
to T
Then if we do this, T,
to T
G,
G
C.
C A C.
Now, let's now do this if it was an M. So C pairs to what? To G. Huh?
G
This G will pair to what? To C. This A will pair to what? To T. But do we have T in mRNA? We don't have. Huh? So that's what we said. Huh? U. Then T pairs to what? A is there. Can you see that? There we don't have U. C, G, A to T, we don't have that. Did I do the correct thing here? Oh, sorry, I lied there. A is there. Why were you quiet? Clear? Then C there. to G, G, A to T, T is not there. Then T to A, A. Is that making sense now? On the base pairing. Are we together? Clear or not clear? Any questions so far? No questions. Now let's look at cell division before we look at the main thing for today. Cell division. What are the two types of cell division in biology? Two types of cell division. Meiosis and mitosis. Meiosis and mitosis. And what is mitosis? Jordan, what is mitosis? You can write that mitosis is a type of cell division that produces two identical daughter cells. Mitosis is a type of nuclear division that produces two identical daughter cells. Is a type of nuclear division that produces two identical daughter cells. Are we together? Two identical daughter cells. That's what happens in asexual reproduction. Clear? In binary fusion. Two identical daughter cells. Are we together? Two identical daughter cells is what happens. So you've got a bacteria here with a nucleus. This nucleus divides into two. And from these two, you form other bacteria, then it continues dividing, and they're exactly identical. That's what you're talking about. Clear? It's also important. That's how we grow. We grow by mitosis. Huh? So growth. Animal growth. We grow by that. Replacing, so uses. I'm looking at uses of mitosis. Replacing of worn out cells. Repairing tissues when your body is repairing tissues is by mitosis. Tissues. Clear. Then the other we said asexual reproduction. Asexual reproduction. And now, if it's so true that we are saying when cell division, each time we are growing, cells are multiplying and the cells are identical. Why is that we have different, you've got an eye, you've got an ear, they're all different. Why is that? If the cell division we're saying each time the cell division is happening, we are saying the genes are identical. So why do we have different parts? Why do you have an ear? Why do you have an eye? Why are they all not the same? What's the reason? What's the reason?
Different cells.
No, it is not different cells, Jordan. We're saying cell division, when you're growing, it happens by mitosis. So the cells are identical.
Yeah.
So what's the reason? The reason is that there are certain alleles that are not expressed. Are we together? Though the cells are identical, there are certain alleles that are not expressed. Are we together? Making sense?
Yeah.
So, what is meiosis? What is meiosis? It is the opposite of this. What is that? Now, meiosis is a type of nuclear division. Meiosis is a type of nuclear division that produces 4 daughter cells. That produces 4 daughter cells that are genetically different. 4 daughter cells that are genetically different. Four daughter cells that are genetically different.
I couldn't hear you. Can you please repeat?
Is a type of nuclear division that produces 4 different cells, 4 daughter cells, sorry. 4 daughter cells that are genetically different.
So you're too fast.
A type of nuclear division that produces 4 daughter cells that are genetically different. Genetically different. Genetically different. So we talk of sexual reproduction. That's why we have an example of meiosis, because we are all different. Sexual reproduction. Clear?
Yes, sir.
Okay. Now, let's look at the main part of this topic here. We talked about alleles, but we want to talk about, so maybe in your family, there are people that are tall. I'm just saying an example. And there are a few people that are short, but maybe you wonder what's happening. So I want to use an example, then we explain as we go. So let's say your mom is tall. So I want to start with what is called, I'll explain, monohybrid.
Should we draw that?
Yes.
That's
So
the top fitting.
yes. So what is known as a phenotype? What is a phenotype? Before we even continue, what is a phenotype? Because I saw people writing funny things in the exam. What is a phenotype? These are what? Phenotype is what? What is a phenotype? These are, yes, outward or visible features. Huh? The outward appearance is what we call a phenotype. So for example, someone is tall. You can see that someone is tall. Huh? So that's a phenotype. Are we together? That's it? Are we together? Genotype is the genetic makeup. If we say genotype, we are talking of the genetic makeup. That's what you call a genotype. Clear? So in this here, let's say capital letter T stands for a dominant. allele. Clear? Two. And let's say small letter t is an allele for dwarf. Clear? Are we together? Follow me. I hope you are writing that down as an example. We are not yet doing any diagram here. We just want to explain certain things that you need to master by now. So, if I write capital letter T and capital letter T, according to that, this is homozygous. Homo means the same. So this is homo because they are the same alleles. So this means this is homozygous toe. Are we together? Homozygous toe. Are we together? Then if I have T and a small letter, what is this? Is this person tall or not tall? Or maybe if we say, what's the difference between dominant, let's go back, and recessive, because we didn't explain this. What's the difference? Who can explain the difference between dominant and recessive? We said capital letter T is for dominant. What does that mean? If something is dominant, what does that mean? Dominant, you need that definition. What does that mean? Who can explain it? Sarah, what do you think? Dominant.
More prominent.
Not really more prominent. If something is dominant, it means that
It's controlling.
yes, what it simply means is that that allele is always expressed in the phenotype. If it's tall, it's always expressed. So that allele is always expressed in the phenotype. Clear? It's always expressed, whether it's homozygous, whether it's heterozygous. So by this, because I've got a dominant T here, this person will still be tall. Clear? Because of this. That's why it's dominant. So it's heterozygous. What is different? We've got a heterozygous tall. Are we together? Heterozygous tall. If an allele is recessive, it can only be expressed in the phenotype when it's homozygous. Clear? It can only be expressed in the phenotype when it's homozygous. So like this here, T, T. That's homozygous. Homozygous. Oh. Are we together? Is that making sense? Okay, let me go back here. If someone who has got a T and a T, this, let's say someone has got a T, capital letter T and the capital letter T, it means this person is tall, but he does not even have a trait of dwarf at all in his genetic makeup. Then if I go back to this person is tall because of this dominant T, but this person has got some traits of what? Of the dwarf in that genetic makeup. But you can't see that in that person. Are we together? Because this is always what? Suppressed. Is that making sense? Then TT means homozygous dwarf. Are we together? Clear? Okay. So let's look at some diagrams. They need to make sense. So if I have a tall person here, okay, and I have another tall, let's say it's male and female that are mating. So now with the diagrams, we'll look at the exam questions, the last 30 minutes for our lesson. Now, phenotype. So this, what can you say about this? We said the capital letter T is for tall. So this person is tall here. This person here is heterozygous, is still tall because of the T. Is that making sense now? Are we together? Then the genotype now. What they normally give you in an exam, they give you something like this. The genotype, you are simply splitting this. So there you've got the T here and the T here. Then here you've got a T and a small what? And a small T. Are we together? Then the offspring genotype. The offspring genotype, you can get it by crossing like this or by doing what is called a Punnett square. Clear? You can actually do a Punnett square there. I'll show you. Hope I've got space here. Now let's start with the one that looks difficult, which is not difficult. So remember, now we have the genes, genotypes. We have splitted this. So this T can combine with this T. Let's say this is the male and this is the female, so that you see the difference. Can now combine with this T and produce a TT. Do you agree? This same T can combine with this here and form a what? A T and a small t. Always start with the capital letter T. Clear? Again, it's a TO, but heterozygous. Is that making sense, everyone? This T here can combine with this T here and form a what? A TT. Is that making sense? Everyone? Then this T here can combine with this T here and form a TT. Huh? Are we agreeing? So what can you say? What comment can you say about the phenotypes? What's this person? Is that person TO? This person TO? This person TO? Because this is not expressed. Huh? Clear. This person TO. So it means you are producing 100% what? 100% TO. Can I write that example there? Are we together? 100% TO. Is that making sense? I hope it's becoming clear now. 100% TO. Now, let's look at another example now. Okay. So let's say now we had male, female. And in this male here, we have heterozygous TO. Heterozygous TO for the female. So phenotype is what you see. This person is TO. This person is TO. And the genotype of parent, normally the right parent genotype is this. So you have your T here. You've got the small t. You've got the T here. For the small t. So by Punnett square, what I meant is something like this here. That's what now I need to show you, which some of you have done that before. So it's up to you. So I can put the male here and the female this side. So if I pick the male here, it's a T and a T. If I pick, I think that's the easiest, the Punnett square. This is a T and a what? A T. So TT produces two capital letter T's. T and that produces T and heterozygous. Then T and that produces a T and a T. And lastly, T, T. Do we agree? Combinations. So offspring. So these are the offspring genotypes. I've used the Punnett square. Who can tell me the ratio of what we have here in terms of the phenotypes? Who can tell me the ratio? Sarah, what do you have here? How many TO? How many dwarf? How many TO? How many are TO?
3.
We have 3. This is TO here. Homozygous TO, Heterozygous toe, remember this is dominant. So 3 toe, 1. So percentage 75%, 25%. Is that clear? Are we together?
Yes, sir.
So now, having said that, we have what is known as test crossing. Test crossing. So, test crossing is done using a recessive. Is done using a homozygous recessive. We use it when we don't know the genotype of the other animal. Are we together? We use it when we don't know the genotype of the other animal or plant. We use a homozygous recessive. So let's say we don't know, let's say we've got your father here. We don't know whether your father is tall. But anyway, we use animals, not people. I'm just giving an example. Okay. Anyway, maybe let's use, instead of people here, let's use rabbits. And let's say the G stands for gray. And the small letter G here stands for white. Clear? So now, let's say I'm given now a rabbit and I don't know, so I'm given a gray rabbit. And I don't know whether this is a homozygous or a heterozygous. The only way I can test that is when I use a white what? A white rabbit. Is that making sense? I'll show you now what we do. Clear? Is when we use, that's why I said we have to use a homozygous recessive. Now, so look at how we do this. So we've got a G, which we said is white. So now, let's say now we're going to work with probabilities, what we're doing. So from what I had shown you, let's say we've got a homozygous like this. If I use my Punnett square like this, and I'll explain certain things. If I use my Punnett square G, small g, I have a G, a G, a G, and a G. So this produces 100% gray what? Gray rabbit. So what it means is that if I use a white rabbit and I met with are we together? I met with this rabbit here that I didn't know, that is GG. I expect, because what can happen in an exam, they can say 98 rabbits after mating were produced and 2 were white. So because of these two, you know, this is probability. So because of these two, you see, this is very close to what? To 100%. So it tells you from the exam, you can now say because 2 were produced, then it's not. No. I want to get that thing. Because now, remember, certain things, mutations can happen. So what it means is that when you've got a very high number like this, close to 100%, it means this is the correct scenario. Are we together? Is that making sense? Now, let's look at the other example. So I'm just talking of matching. If we were matching with one that is heterozygous, that is G like this. Look at your Punnett square. You produce something that is 50%, 50%. If I'm correct. So I've got a G, G. So that with that, you produce a GG. This with this, a GG. So then that, see, Am I correct here? No, I'm wrong. So this is a GG. Because I'm saying this and that. This and that G. Then this and that GG. Then GG. So it's 50% what? White. 50% gray. I hope it's making sense. So now if you had let's say 100 or maybe 40 rabbits were born, then you had 18 white, 22 gray. So this suggests a 50% what? 50%. Is that making sense? Do you have questions? Anyone there who didn't understand it? Are we together? You guys are quiet. I hope you can hear me. So I'm saying this suggests that, because we're doing a test crossing, that this was a heterozygous, that the gray rabbit that we used was a heterozygous one. Are we together? Is that clear?
Yes, sir.
Or not clear? So when we talk of mono, what we are talking about, everything that we're talking about is to do with monohybrid. Because monohybrid inheritance is when you have a dominant allele and a recessive allele. That's what we call, all this what we're talking about here, especially for tau, is what we call monohybrid. But we also have what is known as, so we're talking of monohybrid, we have what is known as co-dominance. Co-dominance is when we have both alleles that are dominant. So they are dominant. Both alleles that are dominant. Both alleles that are dominant. So for example, a very good example is blood groups and the sex cells. So by the sex cells, we'll start with that. A male has got what? And a female, who can tell me the genotypes? You know that from reproduction. A male has got an X and what? And a Y. The reason why they are written in capital letters is because both of them are dominant. There is no recessive one here. Then a female has got X and X, both dominant. So that's what you mean by co-dominance. Both of them are dominant. And if we do crossing here, in terms of your probability, you get X, 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 Y, X, Y. It's always 50% male, 50% female. Because I've seen question papers where they say a family we're having, a family has got 10 daughters and 2 sons, and they're saying what's the probability of them having a daughter? It's not determined by how many daughters, it's determined by this. Clear? It doesn't matter if they have all daughters. The probability that they will have a next daughter is always 50% because it's determined by the genotypes. Are we together? It's always determined, so it's just for you, them to trick you there. It's always determined by the genotypes. Are we together?
Yeah.
Now, let's look at the blood groups. What are the blood groups that you have? Blood groups. What are the blood groups that you have? We have blood group what? A, you know that. Blood group B, and blood group what? O, huh?
Oh,
Do you agree? So in terms of biology, I'll show you what we have. A scenario where we have A, IA, IA like this. So this is homozygous. Homozygous blood group, you can write that blood group A. Then we have a situation where we have A or like this. So the IO, you have to write it down, maybe NB here, is a recessive. So we don't mention that when you're talking of codominance, we don't talk of this. But this is a recessive. In codominance, you don't have a recessive. A and B are codominant. Clear? These two are codominant. So this is recessive. So this is heterozygous. Blood group A. Are we together? What happens if now I have A, since they are both, it results in blood group what? A, B. I didn't want to mention it here. That's the other blood group. We've got four blood groups, clear? Because they are both dominant. Are we together? A, B. So we can also, under B, it means we can have A, B, or we can simply have B and another B. Clear? Making sense?
Yes, sir.
Okay. Now, I want to ask you a simple question. If we were to have a child that has got a blood group O, what are the possible combinations of the parents that can produce a blood group? Which means blood group O, since it's recessive, it can only be this. I'm sure you agree. Otherwise, if it's not that, you can't see the O. The O is recessive, clear. So what are the possible combinations of the parents that can produce this? What are the possible combinations of the parents that can produce that? What do you think?
Blood type O and A.
Yeah, but what combinations? I want the genotypes.
O.
What are the possible combinations that can produce that? Think of the crossing here. Is it possible here? So what are the possible, anyway, before I even tell you the answer, what are the possible combinations of the blood groups? We're talking of the blood groups here. Don't even think of the X and Y. The X and Y is to do with sex or whatever. It's when we're dealing with things that are sex linked. So we'll talk about that. But here, it's the blood group has got nothing to do with sex linkage. Clear. So now, what are the possible combinations that will give us an IO? Possible combinations?
IA and IO.
You said I.
A and IO.
What about here?
With IA and IO.
IA,
the same.
okay, IA and IO, correct. Why? Because I must have an IO. As long as I have an IO allele on both of them, I will have a chance of having this. Is that making sense? Is that making sense?
Yes.
If I have a blood group B here, it must have an IO if I have a blood group A, even if they're different A and B like this. As long as I have this, I will have a chance of having a child with the blood group 1. Is that clear now? Blood group O.
Yes.
Is that clear or not clear?
Yeah.
Clear. What they can also say is, what about if I want 25%? Okay, let me show you what I mean here. So from these combinations here, if we do the Punnett square, if we are quick, sometimes you can actually do it in your head. So now, just to show you certain things here. If I have from a Punnett square, if I have blood group A or blood group B here, sorry, B or here, there I will have AB. I'm sure you can see that. There I'll have blood group B. There I'll have blood group A. Okay, you can do that. There I'll have blood group what? Or. So it means in terms of the phenotype, offspring, I have blood group. So I'm talking of blood groups here. I have blood group A. I have blood group B. I have blood group AB for the first one. I have blood group B. I have blood group A. And I have blood group what? O. Can you see that? Is that making sense, everyone? Can you see that this combination gives us all the blood groups?
Yes.
It gives us 25% AB, 25%, I'm just showing you the B, 25% A, and 25% O. Is that making sense, Sarah? Now, if we use a different combination, as one that was suggested when we started, if I have let's say same, oh, sorry. I have AA here, blood group A. I have another blood group A, heterozygous. I have another blood group A. Then I only have one O. Clear? So this is 75% A and 25% blood group O. Is that clear?
Yes, sir.
Okay, well, you have to play around, especially in multiple choice question. Okay. Then we have what is known as sickle cell anemia. What is sickle cell anemia? Who knows or who remembers?
It's blood type that's genotype resistant to.
Yeah, but I get your point. But so sickle cell anemia is a condition where what happens to you? Because you're talking of the hemoglobin here. Remember, your hemoglobin is disc shaped. Your red blood cells are disc shaped, so that they've got a large surface area for what? For a hemoglobin. So if someone has got sickle cell, you find that it means the hemoglobin here are thin. They are not disc shaped. They've got a small surface area. So it's a condition where the red blood cells here have got a thin surface area. Clear? Sickle cell anemia is a condition where the red blood cells have got a thin, so they're like deformed. It's caused by mutation. It's caused by gene mutation, where the red blood cells have got a thin surface area. Clear? So if they've got a thin surface area, what effect, or how does that affect? If they've got a thin surface area, it means less absorption of what?
Okay.
Hemoglobin is responsible for absorption of oxygen, less absorption of oxygen. So it means you are it's a life risk, yeah? Condition. Clear? Are we together? On sickle cell. Is that clear? Everyone on sickle cell? So, having said that, on sickle cell, now let's use examples. So let's say H here, this H is for hemoglobin, HN, HN. So this is homozygous normal. Homozygous normal. So it means this person is got a normal, he's got no traits of sickle cell. So now, then this with a small n is heterozygous. So it means this person has got some traits of sickle cell. Clear, but they don't have sickle cell. Clear? Are we together? Then NHHN. This is homozygous. So this person here is a sickle cell. Life, you can write in brackets, life-threatening. Huh? Clear? Life-threatening. So most people, we either have this, we either have this. But we're going to talk about something about malaria here and to leave this. Clear? Someone's network here. Now, the malaria, which happens in most places that are warm, like tropical, the malaria parasite, because of the shape of this person with a sickle cell, struggles to penetrate these cells. Are we together? And the advantage of this, although it's life-threatening, is that the malaria parasite struggles to penetrate these cells here. So it means a person who is sickle cell is life-threatening when it comes to oxygen absorption and things like that. But that person can never have malaria. Are we together? Is that making sense? The person that is homozygous like this, that has got sickle cell, can never have malaria because we said these types of cells, malaria parasites cannot penetrate them. Are we together? So it means in the region that is high with malaria is this person here that is at high risk of dying from malaria. Are we together? Making sense? This one has got a 50% chance of surviving. This person here has got a very high risk of dying in a region that it is infected. Is that making sense? You guys are
Yes,
very
sir.
quiet. Okay. Now, we want to look at what is known as pedigree diagrams. Okay, what another bio. Bio. Inheritance. I thought also cancel. Okay, now there. One. Okay, just hold on. I hope you are still all there. All there, all there, all there. Now define the term gene. What did you say the gene is? Who remembers from without looking at your notes? What is a gene? Very easy. We said it's a length of what? You forgotten. A length of DNA that codes for a protein. That codes for a specific. That codes for a specific protein. Are we together? Gene mutation, I didn't talk about that, or I talked about it that now when I was talking about sickle cell. So gene mutation is simply a change in your base sequence. Remember, we're talking about C, whatever, a change in that base sequence. Gene mutation can be positive, can be negative. So gene mutation is a change in base sequence. Change in base sequence of DNA. Are we together? Now, these are known as pedigree diagrams. That's why most of you struggle. So that's what I want to spend more time on the next 30 minutes. Okay. In general, these diagrams here, you find that the squares always represent males and the circles always represent what? Females. Are we together?
Yes.
So the number are represented by B. Small, so if there's a capital letter B, it means this is what? Dominant. Okay. If the mutant allele is represented by a small letter B, so what does that mean? It means this is recessive. It can only be expressed if we have a B. Are we together? Is that making sense?
Yes.
We are losing network. So what's happening with you guys? And someone's network here. So if I go back here, so we have got a female here, a male, with a, so we've got a male and a female here, and it's shaded here. And we are told that, if you read that, hold on. So this is normal, this is normal. Then this is the person with the condition. So if this person has got a condition, yet we said this is recessive. So it means this is B what? B. Do you agree with? Because it's a recessive allele, the mutant. So for that condition to be expressed, it can only be expressed if it's BB. Same for the female here, BD. Are we together there? I'm just explaining certain things here. Then for the normal here, the normal we cannot tell here. We can only tell from this diagram. Then I'll show you just now. We cannot tell whether this person is BB or is BB. Yeah? For both of them, it's possible. Are we together? Then let's go to the diagram. This is what we call generation one. First, the parents. Follow me. I want you to understand the diagram first before we look at anything, because I've seen most of you struggle. So this is the generation one. This is the second generation. This is the third one. Third generation. Maybe this is the fourth generation. Are we together? It's like I'm saying your father, the sons and daughters, then the grandparents, then grandsons. Is that making sense? Everyone? Now,
Yeah.
so this now shows you that this is a couple. Whether they are married or not, it doesn't matter. Anyway, so I'm just saying it might be animals or what. So this is a couple here, this line here. So they are mating and follow this. So it means this here, number 1, is an offspring, because I'm following this line, is an offspring of this. Then number 2, because it's coming from the same node here, is an offspring of this. Okay. Then this one here is also an offspring of this. And this one here. Are we together? Then this here, it simply means this is not an offspring. So maybe this is a female. Sorry, this is a male here. So it means maybe he married this female. I hope it's making sense. Are we together? So now it's like people being brought to your family. Huh? Same with this here and same with that. That making sense? So it's like a son here, a son marries. Then we have another son marries here. Then we've got a daughter here. Nothing happens to this daughter. Then we've got a daughter here. Maybe is married to this guy. Brought into the family. Is that clear now?
Yes, sir.
Okay. Now we want to use biology to use the logic of what we have been doing. So now let's concentrate with the second generation here. So in the second generation here, we said we've got 1, we've got number 2, and we've got this. So we've got 2 sons here and a daughter. But this son here has got a condition of a BB. So the only way the son can have a condition of a BB is when both of them have got traits of a small b here, and a capital letter B. And do you agree? Because if
Yes.
that's not the case, then it's not possible to have a B and a B here. I hope it's making sense. Because because this is a B and a B, it means one of them, one allele was from the father and one was from the mother. That's what we are talking about. Making sense? Then if we go to number 1 here, because they will ask you what number 1 is, what suggests the genotype. Now, if you go to number 1 here, you have also to go back to this, because these 2 are mating and they are producing a son and a daughter. But this daughter here has got a BB. So the fact that the daughter has got a BB, it means for both of them, they have got it like this. Are we together? Remember, they are normal. So they can't be small b, Huh? Are we together? Remember, I said the normal here is either capital letter B or small letter B, heterozygous or homozygous.
Yeah.
Making sense, Sarah? Making
Yes,
sense?
sir.
Okay. Now, if I go to this again, because I'm having number 5, which is a small b and a small b with a condition, what it means, since this is a normal here, it means it's a B and a B. I've got a small b. That's the one that is combining with this to form BB here. Is that making sense? Interesting, huh? Not difficult. Now, let's go to this. They're saying now state the genotypes of the dogs identified as 1, 2, and 3. We've done that. So heterozygous, number 2 here, BB, number 3. Are we together? Is that clear so far? Mike, you're quiet. You're getting it. Now, the geneticists crossed dog number 4 with dog number 5. Now look at this here. This is now what I said, test what? Remember when we said test crossing, huh? Approximately half. Half means what? About 50%. Clear? Had the condition, and 50% didn't have the condition. So you have an idea here. Approximately, we are crossing 4 with 5. What is 5? 5, I think, is 1 with the condition if we go back. So 5 here, we did that is BB. So we are crossing with the recessive. Clear? Number 5 is, they left out something here like this. So it's a small b. Clear? But remember, we don't know this because number 4 here is a normal, but it can be, number 4 is a normal, but it can be a B here, it can be a B. Sorry, it can be a BB. That's what we want to identify. Is that making sense? But what can produce 50%? Is it a BB crossing with this? We've talked about it before. Maybe you can do it in your head. Is it a BB or a B with a small b? What will produce 50%? If it's a BB, we said it will produce O what? O what? If it's 50%, yes, it will be O heterozygous. Huh? If you do that quickly, if you want to prove it, B. We have all normals, huh? B. It doesn't work. It's 100% normal. Clear? So it's a B and a B. Is that making sense? And now they've said Punnett square max for free. So B with that capital letter B, small letter B, B. Clear? Is that clear? So they're saying offspring genotypes. What are the genotypes here that we are producing? B from this. And offsprings, we have what? 2 normal. Then we've got 2 with the conditioner. Clear? Are we together? So it's called test crossing. Making sense now? Or it's not making sense? Now, before we continue, there's something that I've so we talked about inheritance. We're also supposed to talk about, okay, we'll just take about 10 minutes. Variation and selection. Variation and selection. Now, under variation here, we have genotypic variation and phenotypic variation. Genotypic variation and phenotypic variation. What you see, someone is tall, or maybe your eye color is different. Clear? Genotypic variation is only influenced by genes. Genes only. This one by both genes and environment. Because someone being tall can be environment or genes. By environment, it means the food that you eat. Maybe you drink a lot of milk and things like that. Clear? And you get tall. Are we together? Might not be necessarily genes. Are we together? Making sense? Okay. So, under this, we also have what is known as continuous. Who can explain what's the difference? Continuous and discontinuous variation. Who can explain the difference? Continuous and discontinuous. What's the difference? Continuous and discontinuous. Continuous. So this one is only affected by genes. This one, both genes and environment. What is continuous? Continuous. Is height continuous or discontinuous? Maybe you don't know the definition, but you have an idea. Is height of a class continuous or discontinuous?
Continuous.
Class grades continuous. So with continuous, you have a range. That's why we've got the highest, medium. So with continuous here is when we've got a range. Is when we've got a range, intermediates, range because we've got the highest and slower, intermediates. That's what you mean by continuous, e.g. height. It's a continuous variation. Are we together? Shoe size and things like that. Then discontinuous. There is no range, there are no intermediates. That's discontinuous. Is for example, it's either you are tongue rolling or you are not. Are we together? Discontinuous, no intermediates, no range. Is for example. So these are two extremes, discontinuous. So it's either you are tongue rolling or you are not tongue rolling. It's either you've got dimples or you don't have dimples. Are we together? That's discontinuous. And this is only affected by genes. Are we together? Is that making sense? Okay. Is Ella in the lesson? In this bio lesson here? I didn't see her. Okay. Now, let's talk of selection. So we've got what is called natural selection. And we have what is called artificial selection. Natural selection and artificial selection. Does she know the time? I think I get maybe she's using last week's timetable. Oh, I think I forgot to. Tandy, are you able to send the timetable for this week? I think I didn't send the new timetable. Sorry, Ella. Yes, what's your question? So natural selection. Don't write anything. I just want to explain here. Let's say things that happened over this. So let's say people go and live in a desert. Okay. Let's say maybe 20 years ago, people, a certain group or a certain population migrated and went to live in a desert. So because the desert conditions are very hot, it means for the first years, most of them died. Clear? Then, but some of them survived. Why did they survive? Because they developed, maybe their skin, they developed adaptive features. Clear, naturally. Over years, and they became resistant to desert what? To desert conditions. So because they developed resistance to desert conditions, so you've got a male and a female that is resistant, mating and producing offsprings that are resistant to what? To these desert conditions over years. And that's how the population grows by natural selection. Did that make sense? Because natural selection, you are looking at survival of the fittest. The animals that are weak, they die. And the animals that are strong survive. And when the animals that are strong, they mate, and when they mate, they pass on the resistant alleles to the next generation. Is that making sense? And that continues, and the population grows. Are we together? At each stage, the weak ones die. And we call that natural what? Natural selection. Are we together? So in summary, if you want to write notes, you can write that under natural selection, we have variation in population. You can write that we have variation in population where many offsprings are produced. We have variation in population where many offsprings are produced. Third point is that there is competition for resources. If there are animals, competition for resources among its individuals within the population. Competition for resources among its individuals within the population. Competition for resources among its individuals within the population. Are we together? The next point is that there is struggle for survival. Only the fittest survive. There is struggle for survival. Only the fittest survive. Clear? Are we together? Next point is that the fitter individuals pass on their genes and alleles to their offspring. The fitter individuals pass on their genes and alleles to the offspring. Are we together? The fitter individuals pass on their genes and alleles to the offspring. Clear?
Yeah.
Next point, over time, so that happens over time, maybe over so many years, over time, the adaptive features, over time, the adaptive features of a population will change. Over time, the adaptive features of a population will change due to natural selection. Will change due to natural selection. Are we together?
Yeah.
I think it's now very clear, natural selection. Now let's look at artificial selection, which some of you know very well, maybe didn't know that it's called artificial selection. So natural selection, from the word natural, it happens naturally. Artificial, it doesn't happen natural. We are using humans. Huh? Someone is doing the artificial selection. So artificial selection is also known as selective breeding. You can write that down. Artificial selection is also known as selective breeding. Individuals, under that you read that individuals with desirable features are selected by humans. Individuals with desirable, it can be plants, desirable features are selected by humans.
Individuals with what?
with desirable features. The features that you want, desirable features, are selected by humans.
By what?
By humans. For example, it can be plants. You want a plant that survives in desert, and maybe you want a plant that grows very well or things like that, or that produces a high yield, and you want to cross them together. So we are the ones that are selected. That's why I said selected by humans. That's artificial. Then the individuals are crossed together to produce the next generation. The individuals are crossed together to produce the next generation. To produce the next generation. The genes of the desirable characteristics are passed to the next generation. The genes of the desirable characteristics are passed to the next generation. Are passed to the next generation. Hope it's making sense. Are we together?
Yes, sir.
So let's say we have some maize plant. We don't have to write it down. That produces a very high yield. But it only produces a high yield in a high rainfall. And let's say over the years, the climate, because due to climatic change, we're now receiving low rainfall. So it means this can affect the production of maize in a country. So now it means what we now do is we look for maize plants that, for example, that survive under low rainfall. Then we look, because what we want is, now, we now cross these ones with this, so that the alleles for surviving in low rainfall and the alleles for high yield, we are breeding them together. Clear? Which means now under this, now we can still continue having what? Continue having high yield. Are we together? That making sense? Your
Yeah.
GMOs and all that. Okay, now we'll talk about. I hope it's clear. Or not clear. Any questions so far? Dandy, were you able to send your friend the timetable? I don't think she has the timetable. State the genotype of the carnation of plant that is heterozygous for this gene. So let's analyze the question. FN for red flowers. Oh, sorry, FA. FN for white flowers. And what's the keyword here? Co-dominant, which means both of them, that's why the capital letters are what? Are dominant. There is no recessive there. Making sense? So because it's heterozygous, it means I have an allele for red and I have an allele for what? For white. Oh, sorry. FA, according to the person, FN. Do we agree? Genotype. type, not phenotype. Clear? Now, this is where you now need to know your colors. So if you combine the red and the white, what color do you get? Well, they're now asking this. Phenotype is what you see. Yeah? So what's color? Red and white? Tell us your color. Pink, correct. Pink flowers. Is that making sense? Everyone. Now, let's go to the next question, which is not difficult. So parental genotype we are given. Then the gametes, we said we simply split this here. FA, F, capital letter N. Then FA, FA. Are we together? So they gave you space, you see? Sometimes they tell you Punnett square. Even if so, let's do the Punnett square. Then FA, FA. So this one gives us FA, FA. Then this one, FA, FN. Then this one, FA, FA. Do you agree? Then the last one, FA, FN. Are we agreeing? Disagreeing? Making sense? Then the genotypes. So it's simply what we get from your, we have FA, 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 FA. We have FA, FN, FA, FN. So, and what are the, so what is this here? A is for what? A is for red one. Don't forget. Or A is for red. So this is red here. Red flower. Okay, see the right red. What about this here? So we've got one red flower. This is what? Is it one or two? Two red flowers.
Two red.
And what? This is a combination. Two what? Pink, yeah?
Pink.
Flowers. Proportion 50% red, 50% pink. Is that making sense now?
Yeah.
So maybe you get a bio homework today on these diagrams here. Is still in the question in the lesson, so it's very quiet.
Yes.
Okay. Now, sex in cats is determined in the same way as in humans. Complete below the diagram. Okay, complete the diagram below to show how sex is determined in cats. So they're saying it's just the same. Huh? So it means your male is X what? XY. Yeah? Very easy. So if it's XY here, if you combine here, you get X, X. Combine here, X, X. Combine that with that, XY. Combine that with that, XY. Because it's the same. Huh? Clear? So ratio 50% or 50%, 50%. Or otherwise, simply 1:1 in terms of ratio. Are we together? Yes.
Yeah.
1:1. Are we together? Clear? Now there is what we call sex linkage. Sex linkage are to do with alleles that are to do with your, with like this question here that we want to do here. That will be the last question. to do with those are what we call sex linkage, because they are to do with there are traits or alleles that are attached to your sex cells, to your X and Y. Are we together? Like the color in case that we are investigating there. So the gene for the coat color is located on the X, is located on the what? On the X, not on the Y, but on the X. Okay. Capital letter B for black, small letter B for what? For orange. Now, so my question now to you, remember a male is X, Y. A female is what? Is X, X. Do you remember? What I'm trying to say here? Now, is it possible to have a BB on a male? Is it possible?
Yes, sir.
It's not possible. Why? Because a male has only one X. So a male can either have, so before we even read this, I want you to understand this. A male can only either have a B like this, XB with a Y, which is blacker, a black male. Or a male can have a small b like this with a Y, which is simply an orange. Are we together there? Because a male is limited because of this. So it's only the female where we can have a heterozygous, because we can have a BB, BB, we can have a B. It's the same with all sex linked questions like this. Clear? Or a B, yes, BB like this or B like this. Are we together there? That's what I wanted to show you there. Is that making sense? Okay. Now let's look at the pedigree diagram. At least now I have explained that. Now, a female cat can be a mixture of colors, like what you're saying. Okay. So if it's a mixture like this, what does that mean about this female here? Can someone tell me? It means X, capital letter B, X, small letter what? B, yeah? Are we together? So for this here, it's quite obvious. It's an X, B, and then X, small b. Are we together? Then this is an orange male. So who can give me the genotype of this here? Remember, a male is XY. So what's the allele on the X? Is it a small letter B or a capital letter B? B is blacker, remember? And small letter B is what? Is orange. So what did they say? Orange. So this is a small letter B and the Y. That making sense, everyone? On the pedigree, this is actually a very easy pedigree diagram than the other one that we're looking at. I'll also send you some videos on questions to do with this before. Are we together there? So, what about the number 3? Again, is simply an XB, it's a mixture, and then X is female. Clear? Do we agree? This is a male, so it's XY, but is it a small letter B or a capital letter B? Remember, capital letter B is for black. Huh? Capital letter B, because it's blacker. Then this here is black what? Black female. Huh? So what's the black female? Is not a mixture. It's B, X, B. Are we together there? Otherwise, if it's a mixture, are we together? Then this here is your normal male. Sorry, orange, not normal. So X, B, Y. Then this is your female. So it's an orange female. So an orange female, B. So the mixture like that. Are we together? Clear?
Yes.
Everyone. So what I want you to do is I want to give you a homework, but what I want everyone to go through is, can you just go through the 3 videos maybe today after your lessons? Go through I'll send everyone the videos, otherwise you remind me if I forget. I'm going to send you like 3 videos on just on a revision of pedigree diagrams. Clear? Then I'll send you a worksheet homework to work on that. Otherwise, we are actually done with the whole topic. Are we together? On this, maybe we'll look at questions on natural selection in that next time. Okay.